Yeah, so I will talk today about the hierarchical modeling of species communities, which is known as HMSC, and I will illustrate how HMSC works through a case study on booth inhabiting fungi. But before we jump to the scripts, let me first have a few words about HMSC and about joint species distribution modeling. So joint species distribution modeling is a very active line of research nowadays in statistical ecology. And HMSC is one class of the available joint species distribution models. Today, I won't have time to deeply explain what is the mathematical machinery behind this method, neither to go in much detail on the interpretation of many parameter estimates or things that one can do with this method. But if you are interested, I think that the works that I have put in this slide quite nicely wrap what is the state of the art with HMSC, and especially this book that came in 2020. So what is HMSC? HMSC is a multivariate hierarchical generalized linear mixed model fitted in with Bayesian inference. So with HMSC, we can, what the type of data that we can analyze or include in HMSC are the species data, like species occurrence or abundance data on some environmental conditions, like covariates that we have measured across the sampling units where we have recorded the species. We may also include data on the species level traits. And if we have it available, we can also include data on the phylogenetic relationships among the species in our species or occurrence data. So these data that are shown, these matrices that are shown here in orange color. And then from this kind of input, we can estimate all of these parameters that are shown here in the blue ellipse, which I'm not going to go in detail, but I'm showing here that there is a lot that we can estimate and interpret. This is another way of showing what type of input data HMSC can take. So as the simplest case, HMSC can take data on single species and some covariates across some covariate that we may have measured something across the sampling units where we have surveyed the species. So it's possible to do single species distribution modeling with HMSC, but what is really meant for is for doing this type of multivariate species distribution modeling. So we have many species across many sampling units. If we have many species, we can include data on their traits and phylogenetic relationships, and at the same time, we can account for the spatio-temporal context in which the data have been taken. Like if we have some sort of nested hierarchical study design, we can include this type of hierarchical random effects or non-structured random effects, or if we have the coordinates of the sampling units, because we have this kind of uniformly distributed data across the space, is also possible to have that type of structure and therefore then make predictions across sampling units. So the overall workflow of HMSC is shown in this slide by these 5 steps. It consists in 5 steps. The whole when you are running one HMSC analysis, there are like 5 main steps that you really need to follow. There from that you may go to do something else, but these are like the basic 5 steps that one needs to follow to run a general HMSC analysis, and which are the steps that we will actually follow through the scripts in this lecture. So the very first step is to set the model structure and fit in the model. And this is the most critical step of all of them. Because here, when we are setting the model structure, we need to decide what are our environmental coverage, which ones I'm going to include. Am I going to include traits? Yes, no. Do I have phylogenetic data? How I'm going to include the about the spatiotemporal context of the data or the sampling design into the structure of my model. So this is already done in step 1. After the model is set and is fitted, the next step is to check the HMSC convergence. And here we just want to check that is it satisfactory? Has the model converged? If not, we may want to get back to the model fitting phase and change something. And well, this is case study specific, like what might have gone wrong. We need to check. If the HMSC convergence is satisfactory, then we go to evaluate the model fit and to compare the model, the explanatory versus predictive power of the model, so that we can answer the question, okay, how much does my model explain out of the variation in my community data, or is it just modeling noise rather than signal? Is my model over parameterized? This is like what we check in what we call the step 3 of the HMSC workflow. If we are satisfied so far, our model seems to be doing well in the sense of HMSC is good, looks good, and the model fit looks reasonable. Then it's time for the exciting part where we explore the parameter estimates. We can start by just checking the estimates by some standard plots, which I will show later. And here is when we start making some inference from our data. We start to understand what is going on, how the environmental coverage influence our species community, the traits, do they have any effect, and so on and so on. And then the very last step is then that, okay, then we are ready to make some predictions. Now we understand what is going on with this data. Now we are ready to make some predictions. So these steps are all quite nicely explained in this article, in these methods in ecology and evolution article where the HMSC package is introduced. I said that I will show how all of this is done with a real case study. And because I have done so much work with fungi, I will use some real data example with fungi. Surprise. I mean, you don't need to really understand about a fungi to follow the lecture, but I will give some background so that we can a little bit interpret the outputs that we will get. So the data that I will use consists of fruit berry data on fungi that grow on logs. They were recorded, they were surveyed around 200 logs in 12 sites in Finland, and in total more than 600 species were recorded with the fruit berry-based inventories. When the species were recorded, some environmental coverage were measured, both at the log level and also at the site level. So 3 species decay stage, log volume, all which are known to affect these species communities from previous work. And then at the forest site level, what is the naturalness of the forest site by measured with one index. And then we had some traits of the species that were recorded some morphological traits describing how the fruit bodies of these species look like, and also how the spores of these fungi look like. So we have like some hypotheses that perhaps the size of the spore influences the occurrence of these species, or perhaps species with different fruit body types, they like trees of different species, and so on. And finally, because I mean, not always is possible to know what is to get a quantitative phylogenetic tree for our focal species group. So quite often what we do is that we explore the phylogenetic relationships with a phylogenetic or with a taxonomic tree. So then we also gathered information on the different taxonomic levels of each of the species in the data. So all of this data is within the folder that Jody shared with you. So the species CSV, environment, and traits data files, they contain all of this data. So we will feed one HMSC model where our response matrix will be the fungal presence absence at the log level, which we will model with a proved link function, because this is presence absence data. And then as fixed effects, we will include predictors at the log, measured at the log level, the decay stage, the tree species, and the volume of the log, which is not shown here, but we are including it. And because with HMSC it's possible to have like this kind of random effects at different levels, we can also add some site level covariates. So in this case, we are including the naturalness index as a covariate at the site level. As random effects, we can include to account for the nature or the structure of the study design. We include the identity of the logs and the identity of the sites. And as we will also include a phylogenetic, not phylogenetic tree, but a taxonomic tree describing the taxonomic relationships among the species that are in the response matrix. And we will include some traits, the ones that I have shown in the previous slide. So quite a big model where we have lots of fixed effects. We also have the random effects, we have phylogeny, and we have traits. It's not all that we can do with HMSC, but it is quite heavy already. Yeah. So the first step I said that is that we need to set the model structure and that we need to fit the model. So to do so, you can do this in your own computer with the scripts that I have provided. And the very first step is done with script 1, define models, and script 2, fit models. So now we go to R. So we just opened the define models script. If it opens, it will open. Oh my God. I don't know if it's my connection or what it is. It might be. Now, yeah, so let's go to the very beginning of this define models script. And as I said, this script is perhaps like the most critical of all of them. This is like if you want to reuse these scripts to fit your own HMSC model, this one is the one that you will need to modify the most. because the next ones, they have been prepared so that whichever your model structure is, they will run nicely and you will get the standard outputs. But this one is really what you need to do by yourself. So the usual thing that we need to set the working directory. Here we have the set seed, just that the results are reproducible, even if it's not totally necessary here, but it's nice to have the scripts like this. So the package that you need to feed one HMSC model is the package HMSC. And then for this lecture today, we need also the ape package, which we don't need to run an HMSC model, but which we will use to construct the taxonomic tree from the taxonomic levels of the species. So then how we usually work is like, if I go to my folders, to my folder where I have the scripts that I am running today, you see that I have one folder which I call data, and here is where I store all the data that I will use for the lecture today, for fitting an HMSC model. And then you have to create yourself, or the script will create it for you, one folder, one empty folder, which is the models. Here is like where the fitted, unfitted, and fitted models will be stored, which I will explain later why we want that is so. And then HMSC will create also one folder, one results folder, which we want because there are a lot of outputs that we will get, and we want to have everything tidy. So let's get back to the script. So here is where we say that our data are in this data folder, which we have created, and then we have either created it ourselves, or with the next script line, we will create one empty folder, which is called models. So next let's have a look to the data. How does our species data look like? This is really what we have to do before putting everything into the model. Like, I'm telling you that most of the times that we see problems with HMSC, most of the problems arise from these steps, where it's just what we need to do is to check the data, that the dimensions of the data are right, that we have the right type of data, that we don't have like, I mean, if it has to be like a data matrix, that is a matrix, that if it has to be numeric, it's numeric, and so on. So these kind of checks we do with this script. So as I said, there are like over 600 fungal species in the original data, which was collected by one of my colleagues. And if we look at the shape of the data, this is like quite a typical data type, which we analyze with HMSC, which is super sparse data. We have lots of zeros and a few ones. So what do we do with this data? It's a little bit always problematic that when we have like so many species that occur in so few sampling units, the model becomes quite, the parameter estimates become quite uncertain, and quite often we start by leaving some of the most rare species out. And maybe afterwards we do some separate analysis on those species, because it's not that we want to throw out from our analysis those species, but just that the HMSC model works nicely. We take them away, which I will do in some next lines. But now let's have a look to how the covariate data looks like. So this is in the environment CSV data file. Yeah, so this looks good. We have the 3 species, the volume of the log, the decay stage. What is the identity, the site identity, which we want to have here to then add it as a random effect. And we also have ID, which is the log ID, which we will also add as a random effect to the model. So here, what I recommend is that you look at the covariance among the variables, and that you do like a pre-selection of those variables which you feel that are most important given your hypothesis, and which are as little correlated as possible. So here we have done some pre-selection because we know which ones we want based on some previous studies or explorations. But yeah, here we can, let's see if the plot, yeah, we can do this kind of plots and these kind of checks. And what is also important is that we look at the distribution of the covariates. Here, for example, like let's have a look how the volume of the log volume data looks like. So you see that this variable, for example, looks like a little bit bad to be included as a covariate, and that we may want to transform it to include it as a covariate. So this kind of fixes and explorations we do at this point. Going quite slow, my computer. This is still in the previous one. Let's see. Yeah. So now we have like how it looks like when it's log transformed, which is like a much nicer distribution. And so we do this kind of things with the covariates, and also with the decay stage, we do some checks, as we don't want that this single one that is in decay stage 4, we have only one data point for that. So then we unify it with the decay stage 3 so that we get a nicer distribution to be modeled. Like, these are just some hints when you are working with your own data. This is not specific to HMSC. This is general, but the code is here and hopefully is useful. And then we do exactly the same with the trait data, because the trait data is like basically like another type of covariate data. And here you can see that we have also these taxonomic levels that I was talking about, which are included in this data, which we will use soon. Well, we just do these checks. Here, what another place where I find a lot of problems is that HMSC is quite, HMSC needs that the order and the species which you include in the Y matrix, in the species data matrix, are then the same in the trait data matrix. You cannot have like more species or in a different order in the trait data matrix than in the species data matrix. HMSC won't automatically recognize them. So it's important that you check this by yourself. Like, do I have exactly the same species and in the same order in both data files? And it's the same with the covariates. Like, the order of the sampling units, it has to be exactly the same as in the species data, how you have them. So this is like an important check to always have. So here we just see that, yeah, things look good. And then we do this kind of log transformations for those variables that we find a little bit ugly. It's really slow. I don't know how we're gonna do when we have to do something a bit heavier than some histograms. Yeah, so exactly same type of checks. So the next lines of code, they are for this part where I said that, okay, our data is so sparse. We have so many zeros. We have so many species that occur so few times that we don't want this for our HMSC model. So the next lines of code, they are just to throw those away and to have like a reasonable data matrix that we can analyze today reasonably. So here I'm just selecting those species that occur in more than 20 sampling units, and that leads me to have 81 species. And so we will model these 81 species. Now, remember, the species in the data in the species data matrix, they need to be the same as in the trait data matrix. So we need to extract those species from the traits so that we have the same dimensions in both. That is what we do here. And we always check everything. So then, finally, after all of these checks and tidying everything, we are ready to start building our HMSC to set up the model. So first, we start with the random effect structure. So first we create one data frame where we include as columns those variables that we want to include as random effects. So in our case was the log ID and the site ID. And that is what we are doing in this line of code here. And the next is to tell in the HMSC way, in the HMSC language, what are the units of the random effects. So we do it separately for the site and for the log ID using this HMSC random levels function here. So first we tell it in the HMSC language which are the levels for the site random effect, and then we do the same for the log ID random effect. And then, as in any GLM, basically, we need to define what is our formula for the environmental covariates. So here we want to include these environmental covariates. Here, for example, if we would like to have like some interactions between covariates, this is the place where we would do it. And we do the same with the traits. We have a trait formula as well. So we just tell in the HMSC language which are the traits that we want to add to the model. And then I said that we want to add some taxonomic tree. So the next lines of code, they are just to create a taxonomic tree from the taxonomic levels that I showed. So we create something like this, which is maybe a little bit small to show, but it's just the taxonomic relationships among the 81 species that we will model today. Here, this is quite different from a quantitative phylogenetic tree because the branch lengths are the same among every split. But still, when we don't have like that type of quantitative phylogenetic tree, this might be like nice thing that to look at. We may get some insights from our data with this type of tree as well. And finally, we construct the model where we include all of these things that we just constructed with the HMSC function. So here we just need to tell, okay, what is our response matrix? What is the Y matrix? what is our explanatory covariates data, our predictors, which ones are they? We just tell. In which way are we including these predictors in our model? So what is the X formula? What is our trait data? And in the same way, what is the trait formula? Because we can also have with different types of interactions or whatever with our traits. If we have a phylogenetic tree included, which one it is, and what is the link function, or what is the distribution of the data. In our case is the probability function, because we have presence-absence data. And finally, we need to tell what are our random effects, which we prepared in the previous lines of script. Okay, so with this we have set up the model, and then what we recommend is that you save the model, the unfitted model object. And why is that? Because sometimes these models, they take quite long to be fitted, like maybe some weeks, or it depends, depending on your computational resources. And we may want to put our models fitting in a remote computer or in another computer. So you may want to save the unfitted model object, which you will run later. So the next lines of code, they are just for that, to tell that we want to save our unfitted models within the models file. And then we have a couple of lines of a script, which are just to where we are not yet fitting the model, but because we save our big unfitted model object somewhere, we want to know like whether this made any sense or not, whether it will run or not. So then with this couple of script lines, what we do is to check like for a couple of chains whether it runs. And here we see that, well, for a couple of chains iterations, the sampling worked, so we are happy about this. This script took quite long. There are like several scripts, they won't take this long, but this is like the most critical step from all of them. If this has gone nicely, the next scripts will hopefully go nicely as well. So let's go to the second part of the first step to fit in the models. So the script starts as if the scripts are like independent from each other, like we can run the second script without running the first one if we have saved the model. So they are standalone, so we can the first lines, they are just to set the working directory and all of that again. And then we have like this model fitting part. Here you will see that we have like a different number of, that we have decided to have like 4 HMSC chains, and that then we have like this loop that loops over fitting the model for different number of samples and different thinning settings. And why is that? Because before running like a super long chain, which may take very long to do, and then maybe we are at the risk that something went wrong with that, we may want to check things like at the earlier stage, before we are not really checking whether the results make sense, but whether things are running nicely. So we have these lines of code which are prepared so that then there are like first some smaller MCMC runs which we can go on checking as the code goes for a longer and longer chain. So I will show how that it works. But of course, like I have done this model fitting myself in my computer before this lecture, because this takes some hours, and the longest change maybe some days. So I will show some results of already fitted models, but just that you see, like, things work, and this is how it starts and it goes on. So this is how it will look in the end, that we have 4 different thinning. Actually, I only have thinning 1 and 10. Yeah, because these are like the ones that I just did. Like it starts saving this, like the fitted model objects, and then we also save this unfitted models object here before within the models folder, and when we see that this is going well, things are running, and we have already some fitted models, then the next step is just to check to evaluate the model, the MCMC chain convergence. So let's go to do that. So here there is like a not long but semi-long script, which basically you don't need to worry about. Here you can just run everything at once. And why is that? Because this piece of code, it has been prepared so that whichever model structure you have in your first script, whether you have like 3 or 4 random effects, whether they are spatially structured or unstructured random effects, or whether it is temporal data, or whichever you have, like a presence absence or abundance data, or this script will produce the standard outputs. This has been standardized. So I think this is like really good news that this type of scripts exists. So we can just run it all like this, and it will produce some plots. So after it has run, then we go to our folder, we go to our results folder, and because I have done all of this before, instead of going to the results folder, which you may have, I will go to the pre-computed results folder, because here is where I have stored before these results, but they will appear here to you. So what type of plots do we get for the MCMC convergence? So basically it produces this script, this type of violin plots, where what is shown is the potential scale reduction factors. What basically we want is that in our models, this is as close to 1 as possible. So here what we are seeing is that when we have thinning 1 and only 5 posterior samples, these numbers look like horrible. This is something that we really don't want to look at. This is no. When we increase the sample size, things start getting better. This is closer to 1. And then when we increase the length of the chain much more, so that we have thinning 10 and 250 samples, things look like really much better. I would be like quite happy to have this for my parameter estimates with this, for example. And then because we have so many parameters that have been estimated, this file may look quite long, and I won't go through all of them, but basically we have for every parameter this overall plot and then a little bit zoomed plot so that we really look like, okay, is this close to 1? Yes, it is. This is what we want for making a little bit sense of the results we may obtain. So, okay, success. Our MCMC convergence looks good. And it's time for one of the most exciting parts, which is to look at the parameter estimates, like what do our results tell us about. So again, for that, sorry, I skipped one. I got excited. We need to look at the model fit, how the model predictive performance is compared to the explanatory performance. That is like how well my model does predict for those sampling units which are in our data, but which we don't tell the model, like that we partition the data in this test and validation data. How well does my model predict for those sampling units which has not seen the data yet, compared to how the model predicts for the data for the same data that it has done the model fitting. So for that, again, we have standardized scripts, which you can like just run all at once and you will get the results. Here, the only thing that you may want to change is that, I mean, there are like super many ways in which you can look at this predictive performance. Here we are doing it through two-fold cross-validation, but even that is not the only choice. Like here we have the log ID and we have the site ID, so we can do the cross-validation at whichever of the levels that we have included in the model. So here we have the scripts for either for the site or for the log ID. We can do this two-fold validation, cross-validation. So just to show that things work, we can run all at once. I mean, and this can be a little bit computationally heavy because doing this cross-validation, I mean, it needs basically to refit the model. Many times this is one step that may take a little bit of time. So for our case study, let's see how this model fitting part looks like. So it produced 2 files, one for the two-fold cross-validation done at the log level, at the sampling unit level, and one for the done at the site. Which one to choose is up to the study questions, interpretation, or hypothesis of your own study system. Here I just did this to show that there are like this type of choices that one can make, and just to illustrate. So let's go to the log level. So how does this PDF look like? What do we have here, and how do we interpret this? So here we have the predictive power in the y-axis, and here the predictive power in the x-axis. So basically what we don't want is that the explanatory power has much higher values than the predictive power. because that would be a sign of over-parameterization. That's really what we don't want. Almost basically always the predictive power will be lower than the explanatory power because is unavoidable, but it shouldn't be like hugely different, or if you have like high explanatory power but predictive power around 0, then you are in trouble. You want to get back to your model and make some changes there. So here we have 0.25 for the predictive versus 0.17 for the explanatory. And well, there is a little bit of over-parameterization, but not too bad. And how to interpret these numbers, it would call for another full lecture. So I'm not gonna go to explaining like whether we want what type of values we want here, or what is good or what is bad based on absolute values, because what we have learned and what we know is that it's not really the absolute values that we need to look at, even though we tend to do so. But basically that we have like as little difference here as possible is what we want. Okay, now finally, let's move to the exciting part. Let's look at some results. We are satisfied with our model fit. We are satisfied with our HMSC convergence. We think that we have included all relevant covariates and that the structure of the model is good. We are happy. So we go to the next script. There are like 1 million parameter estimates that we can have from HMSC, because we have for every species and every covariate, and then the combination with the traits plus the phylogeny, we will get more parameters. But the good news is that you don't really need to think beforehand which ones you want to look at, because we have prepared this script, which you basically run as a whole, and it produces all the first set of standard HMSC parameter estimate outputs. And these scripts are quite heavily commented, which I think is a good thing, because then, I mean, the first set of results that you will get, the plots don't look nice for maybe publishing or something. So then you can get back to the script and do like this kind of tidying your figures, and maybe taking some information which you may want to visualize differently, but all the information is here. And so that's a good thing, given like all the things that we can get out of this. So then we go to the results folder. And what have we got with this script? So we have got this PDF with the parameter estimates, which is the most important one, and then some Excel files with a lot of parameter estimates, which are just the numeric values for the figures that are within this PDF. And which you may want to then check later on a little bit in more detail, so that they are here ready as Excel files, so that you can do and look whichever is most appropriate for your study system and study questions. So what do we get? So first of all, HMSC does this type of variance partitioning with these horrible colors. where it partitions how much if each of the covariates that we included in this as explanatory variables explains for each of the species that we included in the species data. So here each column is one of the species, each of the species in our data. And different colors are different covariates that we have included in the model. And both random effects and fixed effects, it quantifies the amount of variance explained by each of them simultaneously. So basically how I would interpret this result, what I would see first here is that, okay, the 3 species explains a lot of the variation in the species occurrence of this data set. What 3 species the log is really important determinant of the fungal species. I find like for basically all of them, these 3 species is the most important variable, and then the other variables explain a little bit, and yeah, even the random effects explain quite little compared to what the 3 species explains. So this is the proportion of variance explained by each of the covariates, but we may want to look at the same thing, but in absolute terms for each species. All of these values are stored in those Excel files that I was showing. So this is, yeah, like one could plot this much nicer, and I think not could, but must, because this is terrible. But still is the starting point and the standard output. So here we see that, yeah, actually like these 3 species explains a lot of the variation for all the species in general, and for some species in particular, like it's basically the most important determinant. Then we have like the so-called beta plots that are the beta parameters are the parameters that measure how the species occurrences are influenced by the covariates that we have included in the model. So I'm not gonna go in detail to check at any of these, but I'm gonna just, let's go on with the 3 species story, which is quite nicely shown here, that here, and here in these 3 columns, we have the 3 species. And what we can see is that here we have, for this species here, we have these blue responses. Here we have the red responses. And what do they mean? Is that the species up here are negatively affected by these 3 species, and here positively by these 3 species. So basically these species don't like to occur in these 3 species, and these here, they do like to occur, basically. And if we look at the taxonomic tree, which we included in the data, you will see that actually these responses are quite much, that they match with 2 big clades of species. Because here, basically, we have one clade, and here we have another clade. So these are like 2 well-known fungal groups, classes, Ascomycota here, Basidiomycota here. So we have like quite phylogenetically structured responses. Okay, but how do we know that these are like really phylogenetically structured responses, like from a statistical point of view? Then we have this raw parameter, which informs us about the phylogenetic signal in the data, which informs us like, okay, how phylogenetically, taxonomically structured the species responses are. This value goes from 0 to 1. The closer to 1, the more phylogenetically structured, the closer to zero not. And here we have the posterior probability of this estimate being closer to 1 or to 0. And here we see that is statistically supported, the result that here we have this kind of phylogenetic structure in the data. And then we have gamma plots, which show us how the responses of the species to the environmental covariates are influenced by the species traits. This plot usually looks like this, is usually not like super nice plot to look at, but still we have it here because we can like get deeper into this later on if we like. But I think that the trait responses are best shown as with the predictions that I will show by the last script. And finally, another type of parameter estimates of HMSC are the so-called species-to-species residual associations, like what are the statistical associations among the species pairs once we have accounted for the environmental covariates, for the effects of the environmental covariates, and we can estimate them for each of the random effects levels that we have included in the model. So here we see that at site level, no species associations at all estimated, but we have some at the log level. How to interpret this is like really up to interpretation. Is this because we have forgotten some important covariate to be included in the model, that it would explain some of these species occurrences or co-occurrences? Or is it some hypothesis on species interactions that is like really up to interpretation and speculation? But here they are as standard outputs. And now, after we know what is going on with our data, after we have done like this marathonian work of going through all of those parameter estimates, and we have some idea about how our system works, is really the time for the last task, which is of the last script, which is to make predictions with HMSC. Here there are like 1 million choices about like how exactly you want to do the predictions. But so that you get started, we have also this kind of quite standardized scripts by which you will get kind of predictions for some basic statistics, like how does the species richness, what is the predicted species richness based on this covariate or that covariate that I have included, or what is the relative to traits? Do a species with this trait increase? Does the probability of occurrence increase or decrease with this covariate, and so on? So these scripts creates this kind of a standard prediction outputs, and again you can just run this at 1, and then when you have detected some result that you want to go into more detail, you can get back to this script and extract information, visualize it better, and try to understand it better. But the scripts are quite nicely commented, so that this can be done a little bit to facilitate the work, because here there are like lots of choices that one may want to. And now it's going very slow. I don't know if it's because my computer is tired or this shouldn't take that long. But in any case, let's go to the results folder where I have the pre-computed predictions. So this file is rather large because we have so many cohorts, we have so many traits, and so on that we have so many combinations of variables that we can look at that we may want to make predictions. But in any case, here there are like some standard outputs that I think that they provide a very good starting point to check at some results that may be interesting for you. And of course, we can do the predictions because HMSC makes also this species level parameter estimates. We can look at the very same predictions for every species that is in our data. So here we have 81 species that we included in the analysis. So we could get like 81 plots times as many covariates that we have included in the model. But just to give some examples, so here we have, for example, how the species richness is predicted to change in different tree species. We have some patterns here that birch and spruce, they are the most species rich log types, perhaps not the most exciting result, but it's just to illustrate. And this is for one given species, so that you see that we can predict these very same things for a species, like how the occurrence probability of this one species changes in different log types. So this specific species really likes spruce, like the probability of occurrence of this species is highest in spruce. So we create this kind of bar plots for categorical data. Here we have like a little bit more strong result that the species that have this type of fruit body, they really like this type of conifers. The probability of occurrence is highest in conifers, and so on. But what I wanted to show is that, I said that this is huge. That for continuous covariates, we can have like this kind of scatter plots and the prediction line and the uncertainty of the confident intervals, credible intervals around them. So here we have for every trait, every covariate, this type of plots. And then there are like this type of choices that one may need to make about like whether we go for this type of marginal prediction where if we have like a focal variable, what values do we give for the non-focal variables? Do we set them to their mean values in the data, which is the marginal prediction, or do we go for this kind of absolute total prediction where they are set to the value that they have in the data in reality, so that if this variable is high, when this variable is high, we will show it so in the prediction. And this is why every prediction here is shown twice, because one is for the marginal and the other one is for the total or absolute prediction. And this was a huge monologue, but we went through the whole basic standard HMSC, and I'm sorry it was like quite long, but it's a lot.
Great, thank you so much, Nerea. That was fantastic. So again, you can submit questions on the, let's see, I think Jody had put the link in there, the pollev.com link.
So maybe I stop sharing, right?
It's up to you. I can go through the questions here, and then it may be useful to have some of it up just in case.
Okay.
So again, I'm just doing this. These are ranked. So the top question, or the first question, is in the example, we removed from the database those species with less than 20 presences. Is there a rule of thumb to filter species with few presences? And must such a decision be based on the number of variables and model complexity?
Yeah, this is a very good question. This is like a rather arbitrary choice. I mean, what we ultimately want is like a solid model, like, and we want to add as many rare species as possible, but still that the model is solid. And how to do this, I haven't found like any rule of thumb myself. Sometimes we have used this, let's leave like those species that have less than 5% of the prevalences out, and then things have gone quite nicely. But that is like really arbitrary. Unfortunately I don't have like a good answer for that, because it's really context dependent. As in the question it was put as well, it of course interacts a lot with the model complexity, because the more elements you add to the model, to more parameters to be estimated, the higher risk of over parameterization and uncertainty you are adding. So you need to find the balance based on the knowledge on your own study system. Yeah.
Great, thank you. The next question is, other than accounting for species traits and phylogenies, what are other reasons why a researcher may want to choose HMSC for a joint distribution species model, rather than alternatives like SP occupancy or unmarked?
Yeah, this is a very good question. And I think that I am perhaps the worst person in the world to answer this question, because I have been working so much on HMSC that I have lost a little bit like my view. I mean, if you can do the same thing with another package, I personally like, I don't see like any benefit of using specifically HMSC. Like, but of course, like it depends very much on what type of data you have and what study question you have. We have put a lot of emphasis on trying to have as large species communities as possible, on the fact of being quite flexible with these spatio-temporal structures, which might be a bit, which are quite tricky to implement in the technically speaking, they are quite tricky to implement. And of course, like are in the traits and phylogeny, but I think that actually like one of the main advantages of HMSC is like in adding more and more species, and this spatio-temporal structure part of the model. But I haven't really done like a review myself of what available methods there are, and I know that this is a very active line of research, so all the time like new things are coming, and yeah, it's exciting. Let's see where this line of research gets us.
Great, thank you. I'll just say too, I use SP occupancy and unmarked a lot. I think probably the biggest difference is just sort of the recognition of like how to account for imperfect detection. And so the SP occupancy and unmarked are sort of geared specifically for that kind of data.
Exactly. Yeah. With HMSC, it has not been developed for that type of data. We cannot like really deal with this imperfect detection as well as with that other type of models with HMSC currently. Yeah, which was one of the things that we had in mind, and actually we took some action to implement, but you know, things take time, and yeah.
Oh, that's great.
Yeah, good point that, for example, like you might not be the best choice for that. Absolutely.
Yeah, so I think it probably depends a little bit on the kind of data that you have, how it was collected, and sort of what your objective is, just like you said. But great. So the next question is, can someone run the HMSC model using real data on the relative abundance of species rather than just presence-absence data?
I mean, yeah. I don't know at the moment how this Dirichlet distribution is implemented. I don't think it's yet published, but it's something that that we are working on. But for example, with a typical metabar coding data, which is also a little bit of this type of compositional data. Perhaps not in the best way, but we have published a lot of studies where we analyze this type of data. And I believe that in the book that I showed, we had one example actually with maybe plants with the relative abundances of the plants in the plots. But yeah, not yet perfect, but it's possible.
Fantastic. So the next question is, if you continue to have high PSRF values for some beta values, even after increasing the number of samples, is it okay to consider those beta values unreliable, or is the whole model unreliable?
So here they are asking about one case where we have like many parameter estimates, and these potential scale reduction factors are fine, but then for specific parameters, they are not that fine. Yeah, this can happen. And I mean, it depends a little bit like which are the parameters that have got this type of not very nice, not values that are close to 1. If those parameters are the beta parameters, which are like quite central parameters to interpret the data, then I wouldn't trust the whole model. Like I wouldn't move on from that. But then if it's some parameter which I am not even looking at in my article or in my study, like for example, many times we estimate these omega parameters, but we don't really look at them in detail, and we know that they are quite difficult to estimate to get like to be confident with those parameters, because those matrices are like huge in dimension, and it's tricky. If it would be that case, I wouldn't be that worried. Like then I wouldn't really look at those parameters, and I wouldn't trust those parameters that much. But the model, if otherwise, is fine. I would still trust the model. And something that I often do is that I compare the results with different thinning and different amount of samples. This is like one hint, like compare, are the models, whichever is the number of samples or the thinning, giving me the same overall results. Oh, then I am in a little bit safe territory. Like this is also why we have this script that it goes through these many samples and thinning and so on, so that you can compare. So I would, in addition to looking at this PSRF, I would also look at whether the parameter estimates they are telling me the same story in whichever is the number of samples and thinning. Yeah.
Great. So the next question, sort of that's got the highest number of votes, is what is the best way to account for imperfect detection in HMSC models?
Maybe you can answer this better than me. I honestly, I haven't done this myself. Like, I haven't really been working with mammals or this type of communities where this type of modeling has been really developed. But I mean, if your species community is small and you still have like this kind of repeated surveys, perhaps HMSC is not like your best model choice to start with. There are like other modeling frameworks that will deal with that in a nicer way. I mean, like there is not a standard way to account for the imperfect detection, but if somebody is like super advanced in the technical skills, could in theory do it. It's not that it's not impossible, it's just that we haven't had time to really do it, implement it.
Great. Yeah, and I think there's sort of other packages depending on if your goal from the beginning is to sort of analyze occupancy data, or you have some kind of capture recapture or spatial capture recapture. There's like lots of other sort of packages to do that. But yeah, it sounds like the big advantage of HMSC is when you have a very, very large community. And then I think that probably the unique aspect too is the phylogeny, right? If you have phylogenetic data, that's a little bit harder to incorporate in some of the other packages. So that's a really big benefit there, but fantastic. So the next question then is, can you sort of understand how species interact after removing effects of environmental variables?
Well, I mean, not really. Yeah, this is like an ongoing debate always, like how should we interpret these species-to-species associations that we estimate from the random effects, which are like those associations that remain after we have accounted for the environmental covariates, measured environmental covariates in the data. So in theory, if two species are positively or negatively interacting, in theory, we will either find them, whichever the environmental conditions are, together or separately if they are competing with each other. But that is like a huge assumption to make. And this type of analysis doesn't give you proof about the interaction. That should be interpreted with caution. Like interactions might be the reason why you find those residual species-to-species associations, but they might be like many other things, like covariates that we have not included in the models, or anything that we have not really accounted for. So yeah. We can speculate, but we should be cautious about this interpretation.